Hi, my name is Karen Morin, and my paper, appearing in a forthcoming issue of Antipode, is titled Carceral Space, Prisoners and Animals. My paper hinges together two distinct areas of research: carceral geography and critical animal studies. The overall aim of the paper is to bring forward understandings of the carceral from a broader vantage point than has yet been done. Cross-pollinating carceral geography and critical animal studies allows us to reflect on the ways in which similar forms of violence against human and non-human animals is made possible through the prison industrial complex, the agricultural industrial complex, and the medical industrial complex. Non-human animals in the spaces I discuss are of course not abjected as criminals, yet as property of humans and through various other relationships with them, they become functionally a part of the carceral. The sites and institutions I study include the prison execution chamber and the animal slaughterhouse, sites of laboratory testing of pharmaceuticals and other products on incarcerated humans and captive animals, and sites of exploited prisoner and animal labor. The shared structural conditions at these sites of slaughter, experimentation, and labor are all a part of ordinary, everyday industrialized violence in the United States and elsewhere, and thus a carceral comparison amongst them is appropriate and timely. The oppressions that humans and non-humans share are not the same. The positionalities of the subjects are not the same, but the forms of violence at these sites are the same. Specifically, my analysis focuses on a number of key aspects. First, the emotional, psychological, and physiological trauma enacted on bodies in the day-to-day operations and practices of these carceral sites. I focus on the embodied experiences of men and women incarcerated in regular American prisons and jails, and certain types of sentient land animals who, evidence shows, are among those able to suffer, to feel pain, grief, fear, and sorrow. Second, I study the ways in which animalization of incarcerated bodies works to create conditions for their social death, disposability, and killability. The process of animalization subjugates both certain human and certain non-human animals into hierarchies of worthiness. How the distinction between human and animal is made and unmade through carceral processes is a key feature of my analysis. Third, I examine the geographies of these carceral spaces, their remote or secreted locations, spatial layout and design, and their regulated and technological control features. The geographies of the prison execution chamber and the animal slaughterhouse especially map uncannily well onto one another. At these sites, the killing is divided into stages. Their assembly line procedures are routine and mechanical, highly segregated by task, and out of sight of one another. Finally, I examine relationships between carcerality and purpose breeding that extends across both animal and racialized prisoner populations. We are all aware of purpose-breeding animals for food and biomedical research. Many might argue, though, that incarceration and punishment of prisoners is fundamentally different than captivity and abuse within the slaughterhouse or research lab because prisoners are incarcerated as a result of some wrongdoing, whereas animals are not. And yet we know that the entire apparatus of the prison industrial complex relies on a whole host of social, judicial, and economic policies to ensure criminalization of black bodies that are, effectively then, purpose-bred for prison and who disproportionately end up on death row. Understanding the trans-species violence inherent in these carceral sites offers an important opportunity to challenge their technologies and disciplinary regimes. If you find these topics compelling and would like to ask questions or discuss my work, please feel free to email me at morin@bucknell.edu. Thank you.